脑人言
All right, so welcome everybody to today's new report. This is gonna be the 50 new report preview, and we're gonna honor to have Chilri today here to present her recent work and published on Nature. So it's about the cytosine DNA methylation. It's about the part of the whole brain initiative cell consensus network, BICCN 2.0. So it's a wonderful work, and I'm gonna give a very brief introduction about Chiu Rui. She she graduated from Shanghai Jiaotong University, and now she's at the UC San Diego together with the Salk Institute at the Joe Akers Lab. So let's welcome Chiu Rui. And
Thanks for the introduction. Okay, so today I want to talk about my recent work, which is mainly to build a cell taxonomy and the multi-omic map of the adult mouse brain. And due to the time limit, I will only be covering part of our paper. And if you're interested for more details, please check out our paper online on Nature for more details. So before I dive into more details, I want to give you some brief introductions. So when people study about neuroscience, there are many three levels, from the very basic cell to the neural circuit, and from the neural circuit, then it goes to the behavior. So trying to understand the cellular diversity is a very fundamental goal in the neuroscience, but the cells in the brain is very complex. For instance, the mouse brain alone contains 1 billion neurons, and each neuron contains at least 1,000 synapses. So, in order to despite these complex cell compositions, the interesting thing is these diverse neurons, they contain the exact same genome, but their functions and their shapes are quite different. And taking this cortex layer cell as an example, where the neuron cells are separated into 7 different layers, and neurons in different layers are quite different in the shape and function, and also they project to different parts of the neurons. The reason for this is because the epigenetic is very diverse in the neurons, in the brain neuron cells. Basically, epigenetics is how the gene expression is regulated. And when we talk about epigenome, it could be further separated into 3 different levels. The first is the genome sequence, and further goes to the linear chromatin, such as the histone modification, and then is the most complex 3D genome architectures. And here in this project, we focus on the genome sequence, mainly referring to the DNA methylation. And we also talked about the 3D genome architecture, which is basically describing how the chromatin in the nucleus is folded. It's not randomly folded, but folded for a reason that is very important for gene regulation. And when we talk about DNA methylation, there are 2 types of DNA methylation. The most well-known one is the MCG methylation, which normally has regulatory roles. And we also study about the MCH methylation, where the methylated cytosine is followed not by a G base, but either A or C or T. And both of these forms of methylation has been shown to play a very important role in regulating the gene expression and chromosome structure and nucleosome position in the neurons. So in order to be able to understanding this complex cell taxonomy of the brain, we need to first start to generate this whole mouse brain data sets. And this would require dedicated experimental design thanks to our team who generated this data set. And the pipeline basically starts with the brain dissection, and we isolated nuclei into single nuclei. And in our lab, we were especially interested in the neurons, so we would do a quick neuron sortings. And this is a technique developed by one of the best postdoc in the lab, who is now a professor at UCLA, which basically, like, after bisulfite sequencing, would have very dedicated molecular handling stuff that we will be able to sequencing the masselome of each cells. And despite sequencing for the single cell masselome, we also have the ataxic data set provided by Binger's lab, which is the chromatin accessibility modality. And we also have our own 3C-seq, which is methylation plus chromatin conformation. And last, we also have the RNA data set shared by the Allen Institute and Broad Institute. And we're all together in the BICCN BICCN group. So all our dissections share the exact same brain, and we dissected the brains in the same method. And to give you a more detailed way of how we did the brain dissection, so basically we start with a very a brain like this. This is a mouse brain, and this mouse is P15, so it's the adult mouse. And we then cut the brains from anterior to posterior into slices with the same length. And for each slice, we would further dissect them into different dissection regions. So we have 117 dissection regions in total for the whole mouse brain. And here are the major regions, which is like isocortex, isocortex, hippocampus, thalamus, etc. So here is a pipeline. So after, with this data set, we will be able to get their spatial information. And here is just a quick pipeline to show how we organize these multi-omic data sets. And starting by the masselome-based clustering using the chromosome 100 kb methylation level as the features, we did 3 rounds of iterative clustering. And the goal for iterative clustering is be able to separate the cells into the finest granular resolution. And in total, we get 4,600 clusters for our methylation data sets. And we also did the cross-modality integration between methylation and RNA. This enabled us to annotate these clusters into cell subclasses and borrowed the annotation from the RNA data set. Those Allen Institute people put a lot of efforts trying to annotate the brain cell types. We also integrated with the ATAC data set so that it would be helpful for our downstream regular element analysis. So here is to give you an overview of the cell type composition in the brain regions. And with this light gray UMAP here, it's all the cells in the methylation data set. And you can see for each major regions, the cells located on different parts of this UMAP, suggesting that the cell type composition is actually quite different in the brain regions. And the molecular diversity is very unique in different brain regions as well. And after knowing this cell type taxonomy, we further want to build a multi-omic atlas. And to achieve this, we need to cross-modality integration these 4 data sets. And the algorithm behind this is generally the SURAT integration, but we did some modification. Basically, it starts with dimensional reduction to put data sets into the same space, and using CCA to find their shared variance and find the anchors between data sets, and using these anchors to pull them into the same space. And so here's the result for our integration with 4 modality cells. which is methylation modality and the chromatin structure modality, ATAC modality, and the RNA modality. So for each UMAP here, there are some background light gray cells. For instance, for this methylation data set here, the light gray dots are all the cells from these three other data sets and colored by the dissection regions. So basically you could see that those colored dissection regions in these 4 different figures look quite consistent with each other. And this is the confusion matrix further validated that this integration is working well. Basically, while the methylome clusters and the transcriptome clusters, we iteratively cluster them separately, but after this integration, clusters and clusters will find a clear overlapping with each other. So after this cross-modality integration, we built this multi-omic genome. And here is an example to show that how this works. Taken TLE4 gene as an example, we have 2 different cell types. One is this one. It's purple circulated and this red circulated ones. And this gene is highly expressed in this purple circulated cell subclass. So we use the MCH level as the evaluation for the R expression level, and you can see it's negatively correlated with each other. The darker color suggests for hypo, so it's negatively correlated. And also for the ATAC accessibility, their cis-regular element, if they are more accessible, the MCH level is more hypomethylated. So this suggests this is working well, and as well, this genome track is showing basically the same information. Plus, we also have the chromatin chromatin structure heat map here. So this is a heat map, and it's plotting the genome and genome contacts. So basically, if there are more contacts, the darker the color, and we will see darker color in this domain for the purple circulated cell type. So this suggests that we have built multi-omic genome atlas. And using this complex cell taxonomy and genome analysis, we will be able to explore some interesting questions. One of them is the spatial methylome gradient that we discovered. So interesting, this is an example where this is the axis on the very top of the brain from anterior to posterior. And we would be able to identify 3,000 differentially methylated genes, and their methylation patterns are gradiently changing from anterior to posterior, and as well for the differential methylated regions, which is likely candidate cis-regular element. Their MCG methylation level is also showing this gradient pattern. So the next question is, we wondered if this is only for the methylation, or this pattern also match the spatial transcriptome variabilities. To achieve this, we thought about the new spatial transcriptome technique. One of them is Murfish. Basically, we start with designing 500 gene panel involving spatial method variable genes, and did the cross-modality integration and imputated the methylation nuclei to the spatial location. This imputation is mainly for each methyl nuclei after the KNN neighboring. It will have the 5 nearest neighbors on this space, and we randomly assign one of the Murphy cells coordinates as the methylation nuclei coordinates. And it's very interesting that we find that this result. So the first row is the ground choose colored by the dissection regions color, and here is the imputated methylation nuclei and colored by their by their dissection region colors. And you can see like the first row correlate very well with the downstream rows. This is the accelerator neurons, these are other neurons, and their colors are quite correlating with each other. So this suggests that the DNA alone preserves the spatial information, and it highly correlates with the RNA expression as well. And more interestingly, if we color the cells by their cell subclasses, we find that this would enable us to identify even smaller dissection regions that manual dissection would never be able to achieve, is such as the layer cells. We will be able to separate them. And plotting here, this further demonstrated the multi-omic also works well on the merfish as well. Like this gene, RSGRF12, is highly expressed in the upper layers of the cortex excited neurons. And so are the MCH and MCG level lower, and we would also be able to observe more contacts in the upper layers as well. So this is the spatial part we talked about, and we further went on to build the gene regulatory network. And a very simple model for gene regulation is normally we think that the transcription factors will bind to the cis regulator element, and then it either promote or inhibit the target gene expression. Of course, the real world is not as simple as this, but a general hypothesis that I think we could all agree is if a transcription factor expression level is highly correlated with the target genes, it is very likely that this TF regulates the target gene. So using this logic, we try to identify the gene regulators starting by pairwise correlation. For instance, here is performing the TF and target gene expression level across all the cell subclasses. And the null is the randomly shuffled background. And we will see like we do have some candidacy pairs that is highly correlated with each other. And so do the transcription factor and DMR age will also perform this Pearson correlation. But despite this, we also require that a TF motif is significantly enriched in the DMR to make them age, and also the DMR and target using the same method, which is correlating them. But a very interesting thing is we find if a DMR and the target gene has contacts with each other, they would show even more positively correlation with each other. And after building them pairwisely, we require to give them a final requiring three of them to be part of it, and it's basically we need three to both all of them to be involved in a pairized correlation to be able to call a gene regulator network. After this filtering, we got 10 million ages involving 3 elements, and in total 800 unique transcription factors, 20K genes, and 3000 K DMRs, which is likely the cis regulator element. So here is an example of showing like our logic based on it. So the TF EGR1 and the gene is highly correlated with each other. So this could also be shown in the RNA and the MCH level where it's very positively correlated, this TF and genes. So the a DMR near this gene also show negative correlation with the ATAC accessibility and positive correlation, a negative correlation with the contacts. And so basically this is one of the example age that we show here. So this indicates that this TF might regulate this gene through this DMRs. But of course, for a validation, more wet lab experiments will be needed. But this is the gene regulator network at the whole mouse brain level that we built, and will be a very resourceful for neuroscientists. So the last part is talking about the splicing isoform, where it has been known where like even if it's one gene, it could be differentially expressed because they have different isoforms. And in order to achieve this, we need to integrate the methylation data sets with the SmartSeq data. So SmartSeq data is different than the 10x single-cell RNA data because 10x is mainly sequencing the mRNA from the three part of it. So it's part of the mRNA, while SmartSeq was sequencing the whole mRNA length, enabling us to know the isoform expression. And here example is the NR exon 3 gene, which is a neuronal gene that has been shown to have diverse splicing in the different neurons. And we can see like the alpha isoform of this gene and beta isoform, their expression level are actually quite different in the cell subclasses. And so we wondered if the methylation might have a regulatory role in this isoform splicing. And here, each dot here is a methylated cytosine site, and we are correlating its methylation level to the alpha transcriptome expression levels across all the cell subclasses. And this x-axis is the genome positions on chromosome 2 from the, and the y-axis is showing the Pearson correlation we have. So interestingly, for instance, for the alpha isoform, here is the region 1. It's very near the promoter regions. We find it's very negatively correlated with the alpha isoform expression. So this might suggest that this region 1's methylation level has a regulatory role on the alpha isoform splicing, while this region 2 is more negatively correlated with the beta isoform expression. It's also near the promoter region. So this suggests that the intragenetic epigenetic patterns might regulate the splicing isoforms. And so I'll just do a quick summary of our paper. Basically, we start with building a cell atlas and genome atlas across the whole mouse brain. And by integration with different modalities, we have created a multi-omic brain cell type taxonomy. And by utilizing the Murfish spatial transcriptome, we show that the epigenome spatial diversity aligns with the Murfish datasets. And mainly using the correlation with each other and some machine learning models, we build this gene DMR TF network and predict the transcription factor importance and their downstream targets. And the last figure, we discussed about the intragenetic epigenetic diversity can predict gene isoform heterogeneity. And thanks for our lab members and our collaborators lab, Maga's lab, Binger's lab. And also thanks for my PhD committee. So that's all for my report today. And any questions?